Today I would like to present a paper which title is The Length of Ribosomal Binding Site Spacer Sequence Control the Production Yield are both intracellular and secreted protein in Bacillus subtilis. So this is the first page of the research article. The expression system that we are using has the RBS, which is the common RBS, and have the consensus sequence with most of the bacteria. That is AGG, double AGG, and also the upstream there are two adenosine nucleotide. And the spacer sequence nucleotide in our case is this one. Generally, the nucleotide of spacer sequence are AT rich, but in our case, the spacer region is not AT rich because it also possesses the BAM-H1 site in our spacer region. So as we consider the RBS, so the RBS in our case is the seem good RBS because some studies reported that TAA-GG-AGG is the strong RBS for Bacillus subtilis, although there is a little bit difference. Here the A is present and here the T, but anyway the RBS is good. But we are worrying about the spacer region in our construction system because all of our construction all of our expression system possesses this spacer region, whether that is PhD01, PhD100, or PhD212, as well as we also modified with constituted promoter, that is P43 promoter. So in this study, they explain the importance of spacer region for translation initiation, and they argue that the optimal spacer region is highly crucial and desirable for high for production yield of a target protein. And they evaluate the different spacer region for both intracellular and extracellular protein production in Bacillus subtilis. So first, they use an original vector, which name is PBS-MUL1, which is a promoter HPA. HPA is a constituted promoter and the strong RBS side. The spacer region or sequence in this original vector are poor nucleotide. So they change the spacer sequence from 4 to 12 by adding only adenosine nucleotide. So they check or evaluate the different spacer sequence for the production up to a reporter protein, that is GUS, beta-glucuronidase and GAP, which is actually the derivative of green fluorescence protein. So as you see here, this is the original vector which contain 4 nucleotide spacer region, and as the spacer sequence length increase from 4 to 7, 8, 9, so the yield of protein in case of both GUS and GAP mutant increase. So they achieved about the highest yield in case of spacer length 7 to 9 nucleotide, and production or the yield was 4-fold increase in case of GAP, while 27-fold increase in the case of GUS reporter protein. And there's assay the relative fluorescence as well as the protein assay.
Do you think it is a new finding? Do you think it is a new finding?
Not the strategy is old, but the paper is professor recently published.
What journal?
I think
journal?
microbial cell factory.
7 to 9 base gap sequence, it is just a principle. Everybody know that. That's not a new finding. As you know. Am I wrong?
No, professor, but we also plan to change our spacer length sequence. So that's why I chosen this paper.
Make it short. It's garbage.
In their SDS page, data also correlate with that of the protein assay. Here one question arises that why the US
You can
expression
briefly
is
present this. It is
okay.
garbage paper.
So then they also check their RBS spacer sequence for extracellular protein. And the reporter protein they chosen as XLX1 and catinase, which is abbreviated as CUT. Here they provide 3 signal peptide in their original vector that is present previously, and they found that the highest production of their extracellular protein was achieved in case of PAL for catinase or CUT, and for EXLX, the BS10 signal peptide produced highest